Welcome to Advanced Anatomy and Physiology, and this unit we will be discussing the digestive system. Our agenda today is to review the alimentary canal and the different layers of the alimentary canal, what that consists of, just kind of doing a review from general A&P. We'll review the enteric nervous system, a review of the stomach and the cells and some of the gastric juices that are released and their importance. We'll look at the liver and especially bile in particular, being bile is part of our digestive system and processes as well. And we'll look at the cells of the liver that produce this bile and whatnot. We'll also look at macronutrient digestion, so really looking at fats, proteins, carbohydrate, nucleic acids, and how those macronutrients are digested or broken down with the aid of enzymes, which we'll be looking at as well. And we'll cap it all off with just a real quick overview of the digestive circulation. So a couple terms that you should probably know. Ingestion, this is the introduction of your food into your oral cavity. Propulsion is the movement of the bolus, and once you swallow the food, we no longer call it food. It is called a bolus once you swallow it. So propulsion is the movement of the bolus through your alimentary canal. We have digestion, which is the breakdown of the bolus and specifically the macronutrients. So again, like your proteins, your carbohydrates, nucleic acids, and your fats. And that can be done in two different ways. You have mechanical, which is a physical form of breakdown, so chewing the food, your tongue moving the food around, the stomach mixing the bolus, if you will, back and forth. And then you have your chemical breakdown, which is specifically looking mainly at bile and different types of enzymes and hydrochloric acid that would also start to break down our macronutrients. We have absorption, which is obviously taking those now, what were macronutrients now are going to be micronutrients, the more simplest form of proteins, fats, and carbohydrates and nucleic acids, and absorbing them specifically into the bloodstream so they can circulate throughout our body to be used in different cells. And then what it remains is obviously fecal matter, which then we defecate out through the body. Now our alimentary canal really starts in the esophagus here, and it is anything that the bolus passes through. So your alimentary canal is your esophagus here, your stomach, your small intestinal tract, your large intestinal tract, all the way down to the rectum. The liver, the gallbladder, and the pancreas are what we call accessory organs. The bolus does not pass through them, but they play a really important role in the digestion process. So if we're just kind of looking right now at just the alimentary canal, the esophagus, the stomach, the small and the large intestinal tract, they all have four basic layers. Now, the difference, though, between some of these areas is that some layers may be thicker than other layers in certain organs, if you will. As an example, the stomach is going to have more muscle around it than, let's say, the esophagus. But they all still have these four basic layers. So this should be a review from general A&P, but just quickly go through this. The lumen here is the space that the bolus would pass through. And then you have your very first layer right here, which is the mucosa layer. And this is called mucosa layer because it releases mucus. So just as a reminder, the digestive system is a mucus-lined organ system. And this first layer here is the mucosa layer, which has contact with the bolus and is producing mucus as well. Our next layer is the submucosa, which is right here. And here we are going to find lots of blood vessels, nerves, and the nerve plexuses, and also the lymphatic system as well in green. And so what happens here is the micronutrients will be absorbed through the mucosa layer here and then eventually transported via the blood vessels and the lacteal ducts here, the lymphatic system, throughout your body in the submucosa layer. And on top of the submucosa layer, we have our muscularis layer, which is primarily responsible for movement or propulsion of our bolus through our system. You will notice there is a little bit of muscle, smooth muscle around the mucosa layer as well, but most of the muscle is going to be in this what we call muscularis layer, if you will. And then the very outside layer is the has a few different words for it. It's called the visceral peritoneum. It could be called the adventitia or the serosa. You can see serosa is what they're choosing in this particular picture here, but this is really paper thin on the very outside. So if I were to take you in the cadaver room and show you the outside of any one of these organs that we're talking about, this would be the layer that you would actually see as a serosa layer. So that should be just a quick review of the alimentary canal and its different layers from A and P, but the muscle that is really controlling movement or propulsion of the bolus through is the smooth muscle. And again, this layers will vary throughout depending on what organ we're looking at, but it's ANS controlled, which is the autonomic nervous system. So remember that you don't have control over this nervous system itself, and so this is either the parasympathetic or the sympathetic nervous system, which we're going to get into talking about in just a moment here. But the muscles are arranged differently here. We have some that are longitudinal, some that are circular, as you can see here, and what they're helping with is motility or movement. And one of the terms that we use a lot in here for motility or movement, we used propulsion earlier, but a more specific term is peristalsis. And peristalsis is a form of movement, but it is where small areas of this smooth muscle are contracting at a time to kind of move the bolus to the next section. And then once that bolus gets to the next section, next part of that smooth muscle will contract, so then it again moves once again. The way that I describe it, my analogy was if you had a large Mr. Freezy Pop and you get down to the very end of the Mr. Freezy Pop and you have this ice down at the bottom, without tipping it up, how would you get that chunks of ice to the top where your mouth is? And that you would squeeze one little section at a time, and as you squeeze that little section, that little ice chunk would keep moving to the top until it reached your mouth, right? So peristalsis is very similar to that. Little sections of the alimentary canal are contracting in order for to move this bolus, and so just kind of think of that Mr. Freezy Pop in that sense, if you will. So the nervous system, the enteric nervous system, this is what we would consider the in-house nerve supply of the alimentary canal. There are, as we'll get into this, there are short reflexes and long reflexes. So some of it's all in-house, like the short reflexes, and the long reflexes are also in-house as well, but it's also out of house, if you will, involving the central nervous system. So we'll kind of look at this. So the enteric nervous system can be found, then the neurons themselves, in the submucosal nerve plexus or the myenteric nerve plexus. So if he says location here, if we go back to our picture over here, this is the submucosal layer right here. So this is that submucosal nerve plexus right here in yellow, and then you can see we also have it here in the muscularis, which is the myenteric nerve plexus. So this is part of your enteric nervous system. This is what's going to essentially communicate with our digestive organs. And so we have what we consider to have short reflexes versus long reflexes. So our short reflexes are a response to stimulus that take place within the GI or the alimentary canal, whereas long reflexes can be both. They can be reflexes that are taking place inside or outside of the intrinsic, or the alimentary canal. And so when we're talking about long reflexes, though, when I mean outside, this is really revolving around the central nervous system, and so that would be like your brain or your spinal cord. So I'll give you some examples here in this picture that we see right here. So this picture here, what we're looking at, this brown layer right here, this would be all the layers of the alimentary canal, this light brown, and then there's your lumen where the bolus would be. So you have your internal, if you will, your extrinsic and your short reflexes are found typically within your alimentary canal, and your long reflexes are found both in and out of the nervous system. So that's kind of why we have some of these boxes in the alimentary canal and some of them on the outside, and long is a little bit of both, okay? So what we have happening here, not to make this overly difficult, but you can see our short reflexes here. This is where there's an internal stimulus, and so that's going to then tell different type of receptors that we have, and a couple of them that we'll mention here are chemoreceptors, which are chemical receptors. So examples would be chemicals such as hydrogens, the oxygen, carbon dioxide, anything chemical related. Osmoreceptors is referring to the amount of solutes, and then mechanoreceptors is referring to different types of stretch receptors as well. And so once those receptors are being initiated from some type of a stimulus here, it will continue down through the local intrinsic nerve plexus, which is sometimes called our gut plexus, or in this case they call it the gut brain. But this is your intrinsic nerve plexus, your enteric gut nerve plexus, and therefore that will tell the effectors. In this case, it could be smooth muscle around the alimentary canal or different types of glands to become more activated or be inhibited depending on the situation here. And the bottom line is it's going to change the contractility or the secretory activity of our digestive system. Now, on the other hand, if it's an external stimulus, such as you see something like food, or you smell food, or you taste food, or even just the thought of food, is going to send information to your central nervous system, and that's going to send information to your local nervous system in this enteric or gut nervous reflex right here, which will stimulate the smooth muscles of the glands to prepare the digestive system for the fact that you're seeing food, or you're smelling, or you're tasting, or you're even thinking about having lunch. It really kind of, what I would say, primes the nervous system. Now, you can see here, one last little thing, you can see that the chemo, osmo, and mechanoreceptors can also go up the long way here and tell the central nervous system that we're having changes in our pH, or changes in our solute content, or changes in our stretch receptors, and the central nervous system can then turn around and send information back down through the local enteric or gut reflex, and then again back through the effector or the smooth muscles. So you have an external stimulus. Just to summarize, you could have an external stimuli, you could have an internal stimuli. So an example of an internal stimuli, because they don't give you one here, would be distension of the stomach as an example. That would follow under mechanoreceptors. They're detecting the stretch in the stomach, and so therefore it would send it through the local, or send it up through the nervous system the long way, and the smooth muscles around the stomach might be affected by maybe mixing or contracting more, and therefore trying to move that food or the bolus, because the muscle or the stomach has been stretched so much. So just kind of an example of how that might be used in regards to that. So really, when we look at this, the purpose of this internal house called the enteric nervous system, if you will, is to regulate digestive activity such as peristalsis, so again the movement of the bolus through, or the mixing portion such as in the stomach. Also gland secretion, whether it's going to inhibit it or stimulate more gland secretion. And then finally the reflexes, in regards to the long reflexes needed for this, so the sight, smell, and taste, and how that's priming our digestive system, getting it ready for the introduction of that food. And so, when you're approaching lunchtime and you're thinking about food, or you're smelling food, your digestive enzymes and hydrogen ions, and there's changes in your pH, are all priming your body and getting it ready for this introduction of this food that it's anticipating. And this is all part of this, what we call enteric nervous system. Now, the enteric nervous system is affected by the autonomic nervous system. This is what's kind of controlling it, if you will, which we have the sympathetic and we have the parasympathetic. So sympathetic, this is our fight or flight situation. So typically we're not in this situation most of the time, right? This is kind of your fight or flight, your scared, threatened situation. But if you are in a situation where you're in the sympathetic nervous system, maybe you're really nervous about something, you're giving a speech or something like that, really what this does is this inhibits muscle contraction. It really prevents the muscles from digestion, if you will. The secretions are inhibited as well because it doesn't want to do the rest and digest stage, if you will. And the blood is shunted away from the digestive system, so decreasing blood flow because the blood has to go to where you need it the most. So as an example, you might have more blood flow to the heart and the lungs and the muscles in a very threatening situation, and definitely the blood flow doesn't need to go to the digestive system at that time. On the other hand, most of us are in the parasympathetic nervous division, which is the rest and digest stage. And so the vagus nerve, cranial nerve number 10, plays a really big role in this. And it increases, parasympathetic would just do the opposite. It increases digestive activity, it increases secretion and muscle contraction and movement and blood flow as well. So I was telling you about those receptors, and these are our major receptors. So chemoreceptors, which are more chemical related, again, like hydrogens, and which is a big one, or bicarbonate ion. Osmoreceptors are the solute concentration and how concentrated the fluids are. And then mechanoreceptors are your stretch receptors. And so remember, going back to our general A&P, our homeostasis is all about the receptor communicating with the control center, communicating with the effector to maintain balance here. And so these receptors are kind of the front line. They're the first ones to experience the changes, and they have to communicate locally to the digestive system, like with that enteric nervous system, the short reflexes, if you will, or they have to go through the long reflexes through your central nervous system, and eventually to the effector to change the response. Okay, so if your pH is too high or too low, somewhere down the line, I won't get into that, but somewhere down the line we have to tell the control center, and eventually something has to correct that response, if you will. Now, our oral cavity has a few different components to it. Obviously, we have a lot of salivation there, or saliva, and the purpose of the saliva is to really moisten up the food before you swallow it, so it makes swallowing easier. But also, there are some enzymes that are located in the saliva which help begin the breakdown process of your food. Now, obviously, not very much is going to be broken down by these enzymes, being that the food isn't in your oral cavity for very long, but I think they say like 10% of your food is broken down by these enzymes. But a couple things that you would want to know about saliva as far as what is the components of it. Obviously, a lot of water is a component of saliva. You have antibodies, which are a good thing that help if in case there's any type of foreign material in there. Mucin, which when combined with water becomes a very thick mucus. So mucin, M-U-C-I-N, combines with water to become a very thick mucus. A couple enzymes you need to know: amylase, A-M-Y-L-A-S-E. You'll see that a little later in the presentation. This helps with the breakdown of carbohydrates. And then you have lipase, L-I-P-A-S-E. This helps with the breakdown of fat. And so we have some enzymes, we have some antibodies, we have some mucus that's being created, we have a lot of water, and the salivary glands are controlled by the autonomic nervous system, parasympathetic versus sympathetic. So when you're in the parasympathetic nervous system, you have saliva easily flowing out of these salivary glands, and when you're in the sympathetic nervous division, that shuts it off, and there is very little saliva being release, and that's why you get that cotton mouth when you're very nervous. And so just to review, there's your parotid salivary gland, your submandibular gland, and your sublingual gland. All three of these are releasing saliva into the oral cavity. In the oral cavity, we also have mastication, which again is the mechanical breakdown of food, and that's obviously to make it easier to swallow the food. That's the purpose. And deglutition, which is which is actually the swallowing reflex. And so we need this in order to swallow our food, and it comes in 3 different phases, which we're going to review right now. So deglutition, which is swallowing reflex, it has 3 phases: voluntary, pharyngeal, and the esophageal reflex. So the first phase, voluntary, is exactly what it says. It's voluntary. It's moving, it's swallowing your food and then becoming the bolus. And so this is very voluntary. You don't have to swallow your food. You can spit it out. But then once you swallow it, though, you go into a reflex, which is an automatic response, an unconscious automatic response of this. Our first reflex is the pharyngeal phase, or the pharyngeal reflex. And then it goes into the esophageal phase, which is the esophageal reflex. So I want to show you this picture right here, because this is the phases that you would want to know. So what we have here is, we have in yellow, this is our bolus, that we're starting to swallow it right here. And if we go over to number 1, we start to see the esophageal reflex taking place here. And so remember, it's an unconscious decision what happens here. And one of the things that happens is the soft palate and the uvula move upwards, as you can see right here. And the purpose of that is to block the bolus from going up in and around into your nasal cavity, if you will, and allowing the bolus to move down down into the esophagus here. The other thing you're going to notice is the epiglottis here is going to also move down, and that's a normal response, and allowing the bolus to go down your esophagus and not down your larynx, which is your windpipe. So then once it gets down into your near your esophagus here, there is a little sphincter up at the top called the upper esophageal sphincter here, which is a muscle that must relax in order for this bolus to get into your esophagus. There it will travel down your esophagus, and as you see here, it takes about 9 seconds for that to travel down roughly, and it's doing that via peristaltic contraction. So again, remember the Mr. Freezy Pop idea. That's what's happening. Little sections at a time are contracting, allowing that bolus to move further down to your stomach. So there are a couple things that you need to know about this. Obviously, is what's happening during the esophageal reflex. What are the different things that are happening with the soft palate and the uvula and the epiglottis? And then once we get to the pharyngeal phase, with the upper esophageal sphincter relaxing and allowing that bolus to move through the esophagus, if you will. Okay, then we move into the stomach here, and the stomach, its purpose is to mix our bolus back and forth to mechanically break it down. It is very acidic down in the stomach. It's about a pH of about a 2, so it's pretty acidic down there. There is a lot of gastric juices down there as well, which we'll look at in just a little bit, that help with some of the breakdown of our bolus and some other components of it, which we'll get to in just a moment. But there are some reflexes that also take place here, and there's 3 of them that you need to know. Enterogastric, which is when the stretching of the duodenum occurs. So remember, the duodenum is after the stomach. Duodenum is our very first part of our small intestinal tract. And so once this stretching of the duodenum takes place after the bolus moves through the stomach into the duodenum, we get this reflex where the duodenum stretches, and it prevents any more acid from being produced in the stomach, because that bolus has now moved out of the stomach, and there's no real need for the stomach to keep producing this acid. So this is the enterogastric reflex that takes place. The gastroileal, and so this is when food is introduced into the stomach. The ileum, which is the very last part of your small intestinal tract, begins peristalsis, and the ileocecal valve, which is the valve or the sphincter between the ileum and the cecum, which is your first part of your large intestinal tract, open up. And this is doing so because being that the food is stretching the stomach, the lower parts of the small intestinal tract are trying to move things along, if you will, because we know that it knows that the more food or bolus is coming its way. And so we start to get more activity in the lower part of the small intestinal tract with this peristalsis and the sphincter, the valve, opening up. and allowing things to start moving into the large intestinal tract. Gastrocoliac, this is where we get peristalsis of the colon or the large intestinal tract, and this is when food is present in the stomach. So again, kind of the same concept. When food is present in the stomach, we start to get more movement in the colon or the large intestinal tract as well, which eventually leads to the defecation process. And defecation really should happen once a day, technically, under normal conditions. Of course, that's not always the case, but if everything was in a perfect world, that's what we should see. We should see every day that you should have a normal bowel movement with this defecation and introduction of food. Now on the opposite end, we have vomiting, which is known as emesis, which is a reflex. It's part of the what we call the emetic center, which is the medulla oblongata. So it's a reflex that automatically happens. We get a lot of pressure that builds up. We get all of our sphincters that are in our alimentary canal to relax. The soft palate blocks the nasal cavity. So just think about swallowing in reverse. Okay, swallowing, we had all those deglutition or swallowing. We had all those sphincters relaxing and the soft palate blocking, and just think of that happening again, but instead the bolus moving out the opposite direction. So the result of vomiting, though, is to try to attempt to create equilibrium within the stomach or the the alimentary canal. With vomiting, though, you could have, of course, dehydration, and because there's so much acid that's being thrown up or that you're vomiting, that you could lead to what we call metabolic alkalosis, which is the lack of hydrogens. You have more bicarbonate, if you will, in your system, and that could lead to an unbalanced equilibrium in regards to metabolic alkalosis. And I don't want to get into too much of that because we'll get into that in another unit later in the semester. Now the stomach has what we call gastric pits, and within the gastric pits we have several different types of cells. So here you can see this is the liner of the stomach here, and these are the gastric pits that go all the way down. And you can see that the gastric pits are lined with different types of cells. So look over here. Here are our 4 layers again of our alimentary canal. There's our mucosa layer, which is where the gastric pits are located. Underneath that you have the submucosa, which are where your nerves and your blood supply are. Then you have your muscularis right here, and this is pretty thick. And then you have your very thin outside layer, which is your serosa adventitia or visceral peritoneum. What we want to concentrate on are these gastric pits right here, and what are these cells that we see here, and what do they do? What is their function? So our first cell, which is this large blue one right here, this is called the parietal cell, and its job is to produce hydrochloric acid along with another protein called intrinsic factor. So we'll get into that in just a moment. Then we have our chief cells, which are in purple here, and their role is to produce a chemical called pepsinogen, which I'll show you and talk about that in a minute as well. Pepsinogen, you can see that right there. You can see hydrochloric acid. The only thing you don't see here is intrinsic factor, but intrinsic factor is coming from, again, the parietal cell here. And then you have your enteroendocrine cells in green right here, and their job is to produce what we call paracrines. And paracrines or paracrines, if you will, some examples would be what they are is just local hormones. They only are hormones that act locally. So paracrines are very loosely we say hormones, because hormones typically are supposed to circulate through our whole body, but paracrines are hormones because they tell cells what to do, but only locally. So just think about like the way I kind of look at this is like in regards to a hormone, it can circulate wherever in your body. So look at like Walmart as an example. It can set up a Walmart anywhere in the world, right? It just has that ability to do that. That would be like a hormone. But a small mom and pop shop can't do that. They stay very local, and that's what a paracrine is. It's a hormone that stays local. Some examples would be histamine, and one that you'll probably see a little bit more is called gastrin. Okay, and then finally you have mucous neck cells that are way up here. You don't see them on this one right here. Mucous neck cells right here. And their job is to produce what you would expect, mucus, because the mucus needs to mix with the gastric juices here because we don't want the environment to be overly acidic. That could be harmful, but also what the mucus does is it coats the liner of the stomach so the acid doesn't touch the cells and start to break down the tissue, because being that it is so acidic, if we didn't have this mucus coat along the stomach, we would get the breakdown of the tissue. So here it again, we have hydrochloric acid, we have mucus, we have intrinsic factor. Now what does intrinsic factor do? What this does is it helps with vitamin B12 absorption. Very important that you know this, vitamin B12 absorption. And what does vitamin B12 do for us? It helps with red blood cell production. So if we didn't have intrinsic factor, we wouldn't have vitamin B12 absorption. We would see a lack in red blood cell production, which in return would make us anemic. And we'll talk more about that in the cardiovascular system than now, but I do want you to know what is its purpose today. Pepsinogen is produced from those chief cells. And when pepsinogen is in contact with the hydrochloric acid, pepsinogen then becomes this, which is called pepsin. So this is why we have pepsin, pepsinogen, or pepsinogen, pepsin. If you go back, you can see that right here. Here's pepsinogen. It has contact with hydrochloric acid. It then becomes pepsin. And what does pepsin do? Pepsin does, it helps with protein digestion. We talked about those paracrines, those local hormones. There was another one I didn't mention, but it's not overly important, but it's serotonin. But gastrin is probably the biggest one there that we'll probably see. Obviously, there's bacteria in your stomach. That's normal. You have undigested food, or the bolus, and you have plenty of water as well. Now, the question that we have is, what inhibits or stimulates gastric juices? What's going to cause them to overproduce? What's going to cause them to underproduce in certain situations? So when we look at this, there are three areas that are going to stimulate or inhibit our gastric juices. One, we have our cephalic, so our brain, our eyes, our nose, our sights, our smells, our senses, okay? We have our gastric phase, which is our stomach, things that will happen in our stomach to stimulate or inhibit, and intestinal, which will have stimulatory or inhibitory effects as well. So let's look at stimulatory first, and then we'll go over here and look at things that would inhibit it. So cephalic is pretty easy. Cephalic means head. So thinking about the sight or thought or the smell or the taste of food tells our cerebral cortex of our brain and the medulla oblongata to activate our vagus nerve, which tells these cells that we were just talking about to start releasing more of these gastric juices. Okay, it's priming the body, right? It's stimulating it, or stimulating the stomach, if you will. Now, when the stomach starts to distend because of the stretch receptors, and this would be both short and long reflexes like we talked about earlier, that too will stimulate gastric juices. Food chemicals and the rising pH activate the chemoreceptors, which tell the G cells, which you don't really need to know, but these are the cells that produce gastrin, that local hormone, to begin to produce these digestive juices as well. And in the intestinal phase, the presence of partially digested foods in the duodenum and the distension of the duodenum when the stomach begins to empty will cause the gastrin release into the blood, and also we get a little bit of gastric juices that are released as well. Very brief though in this one. Now, so you can see gastrin is popping up quite a bit, and that's why I think this is a local hormone that's locally telling these cells to produce these extra gastric juices in the need. Okay. Now, inhibitory, what would prevent gastric juices from being secreted? Well, loss of appetite and depression. This would decrease the parasympathetic nervous division, which is, remember, our rest and digest phase, so that would decrease gastric juices. Excessive acidity in the stomach or emotional disturbances. So excessive acidity would tell the G cells to stop producing gastrin, which we know if we produce gastrin, we typically will tell the cells to make more gastric juices. So with a decrease in gastrin, we decrease our gastric juices. If we're in emotional stress, you're very nervous about something, your sympathetic nervous system is activated, which would decrease the gastric juices. Distension of the duodenum, presence of fatty acid, fat or acidic or hypertonic chyme. Hypertonic would be excessive solutes in the chyme. This would be the enterogastric reflex, both short and long, and the release of enterogastrones, secretin and cholecystokinin. You do not need to know those. You won't see those on the test, but you know, this will start to decrease the activity of the stomach as well. So you can see some differences between what things that could stimulate the stomach gastric juices and what could prevent it. Now, we also want to look at hydrochloric acid production here, and how does that actually occur. And so you can see this picture looks a little daunting, but hopefully it shouldn't be too bad here. So here's how we produce hydrochloric acid. So you have, this is our parietal cell right here in blue, and this is the one that's responsible for producing the hydrochloric acid. And we have a good blood supply here as well. You get CO2 that comes into the parietal cell and it mixes with water. And what happens then is that mixing of the CO2 and the water is combined together by an enzyme called carbonic anhydrase, which produces carbonic acid, H2CO3. Now, being an acid, it has to release a hydrogen. So as you see down here, it releases one hydrogen, and that moves out of the cell. This chemical right here that's left over is called bicarbonate, and bicarbonate leaves the parietal cell back into your bloodstream. Out here we have chloride, which comes into the parietal cell and is pumped out, and this combination of hydrogen and chloride here make your hydrochloric acid. Okay, so you can see that it really starts with CO2 here and water that starts this process and pumping this extra hydrogen out. The bicarbonate goes back into the bloodstream, and chloride moves its way through the parietal cell and out to combine with this hydrochloric acid. Now, one other quick thing here, in order for this hydrogen to get out, it has to go through this hydrogen-potassium pump called the hydrogen-potassium ATPase, if you will, right here. And this exchanges the hydrogen out, and it allows a potassium to come back in. So this is an exchange pump, if you will, in order for this hydrogen to get out. And this chloride right here is actually coming from the blood. It doesn't show you that, but it is coming from the blood here. So review this, review the chemical combination of things that are happening here, what would combine with what to make what, and how do we get that hydrogen out, where does that chloride come from, and kind of go from there. Okay, now what about the liver? This is also an important organ of the digestive system. The biggest one that we're going to talk about, though, and concentrate on is bile production. And we'll talk more about that in a moment. Other things that the liver does that may not be necessarily part of the digestive system directly would be like clotting factors that help with clotting of our blood. The liver helps store vitamins, glycogen, which is a stored form of glucose, and iron. It controls the production of cholesterol and removes bacteria from the bloodstream. It's like a detoxifier, if you will. We probably have all seen the liver before. It has a rather smaller left lobe, a larger right lobe, if you will, and tucked underneath it is the gallbladder right here. And if we opened up the liver a little bit, we would see what we call these liver lobules, these large kind of hexagon type of looking structures right here. And you can see that there are some bile canals. We have a very, very strong cardiovascular presence in here. And amongst many other things, we'll talk probably more about this in lab. We actually look at this liver lobule a little bit more specifically in lab and kind of some of the finer details of it. But the cells right here, these little brown cells, these are called hepatocytes, and they're found in the liver, and what they're responsible for is many different things, but the one big thing is bile. And so if we go here, hepatocytes are found in the liver, but bile is produced in the liver from these hepatocytes and stored in the gallbladder. And what is the composition of bile? It's color-wise, it's a yellow-green color. That is because of bilirubin, which is the breakdown of red blood cells. It is rather alkaline in its chemical composition. It has bile salts, it has the bile pigments, which give it color. There is cholesterol, there is triglycerides, which is a form of a fat, both cholesterol and triglycerides. Phospholipids is a form of fat, so it's a pretty fatty substance. Electrolytes as well. And what the purpose of bile is to emulsify fat or break down fat, not taking fat and breaking it down into micronutrients, but taking a fat globule, as you can see right here, and once the bile salts start to work on it, it breaks them down into smaller little fat droplets, if you will. Now, these fat droplets, if you will, are going to be further broken down into free fatty acids and monoglycerides, because that's what fats are. Okay, they're fatty acids and they're a monoglyceride backbone, and a triglyceride is that glycerol backbone with 3 fatty acids. So if we break down this fat, if you will, we get these fatty acids and monoglycerides and bile salts, which we call a micelle. So a micelle right here, these much smaller little pieces right here, are that of a fatty acid, a monoglyceride, and bile salts all together. And then what happens is the fatty acids and the monoglycerides will be broken down further and transported across the intestinal tract wall and absorbed through the lacteal ducts right here. Now, the fatty acids and the monoglycerides, as they're being transported across, as you can see, they break down even further. These are called chylomicrons. Okay, so we have up here, we have our large fat. It gets broken down into smaller fat droplets because of the bile salts. They get further broken down into fatty acids, monoglycerides, and bile salts, which we call micelle. And they get broken down as they're traveling through the intestinal wall into fatty acids and monoglycerides, and we call these chylomicrons, which are then absorbed in the lacteal duct. So we started with this macronutrient way up here, this big fat, and we ended up with our very, very micronutrients down here. And so this is the emulsification steps that happen. It's not simply that bile just breaks down fat. It's a little bit more detail that you need to know here in regards to that. Now, this video I'm about to show you is very basic, but I just think a visual is also really good to see. So,
what is bile? Bile is a digestive juice that is produced in the liver and stored in the gallbladder. Bile's major function is that it is involved with fat digestion and absorption in the intestinal tract. So let's take a very basic look at how this happens. Here we have Jason, and he has just eaten a meal, and part of that meal contains some fat. And fat does not dissolve in water, so this means his body has to digest it in a different way than it would protein or carbohydrates. As fat moves through the digestive system, it mainly remains clumped together. When fat reaches the small intestine, it basically is large fat droplets. And this is where bile does its job. Bile is released into the small intestine, and it is able to mix with the large fat droplets. Bile contains bile salts, which allow it to emulsify the fat, or break it down into smaller droplets. Next, the enzyme pancreatic lipase comes in and breaks the fat into free fatty acids and monoglycerides. These free fatty acids and monoglycerides are now small enough to pass through the epithelial cell layer that covers the villi. Villi are tiny finger-like projections that cover the interior of the small intestinal wall. Inside these villi are blood capillaries and lymph capillaries. Here, fats are absorbed into the lymph capillary and transported to the liver by way of the lymphatic duct and circulatory system. So as you can see, bile has a very important role in fat digestion and absorption. But bile does a couple of other things as well. It helps remove a substance called bilirubin, which is made when the body breaks down old blood cells. Bile also helps remove cholesterol, as some cholesterol is converted to bile acids and eliminated in bile and feces. And that be the wonderful world of bile.
Okay, so with the video though, you can see that they didn't really go into the terms micelle and chylomicrons, which of course is what we want you to know. But so just be mindful of that. It is a little bit detailed, so you don't want to overlook that and just think that we're just going to ask you kind of some basic stuff. I want you to understand, you know, what would these steps be if we were kind of moving down this bile emulsification process here. Now bile, I forgot to mention, bile is secreted down through the common bile duct from the liver and the gallbladder, because remember, the liver makes bile, the gallbladder stores it. It passes through this common bile duct, which then is secreted into the duodenum. So most of our emulsification is happening in the duodenum, which is the very first part of our small intestinal tract. Now the pancreas is a very important endocrine gland, but also an important exocrine gland. So we're going to concentrate mainly on the exocrine portion. I'm going to briefly talk about endocrine, which is, when we think about endocrine, we're talking about hormones. So the alpha cells of the pancreas as an endocrine gland produce glucagon, which is going to help raise your blood sugar. And the beta cells of the pancreas produce insulin, which lower your blood sugars. And that's part of what the pancreas does. That's not what we want to concentrate on right now. We want to concentrate on as an exocrine gland, which means that it releases its materials or its juices into the digestive system. Endocrine hormones, those were circulating throughout the body. Exocrine is being released into the digestive system. So our pancreatic juices are coming from the acini cells and cells of the ducts of the pancreas, and the pH of this juice is about a pH of an 8. So the question is why? What happens is bicarbonate released via these pancreatic juices, they help neutralize the stomach contents, because remember, the stomach has a pH of about a 2. So when the stomach contents that we call chyme leaves the stomach and gets into the duodenum, it's really, really acidic. And so what the pancreas does is it releases their pancreatic juice, which has a pH of about an 8, which really helps to offset the pH so that when it's in the duodenum, it's a good pH for the enzymes to start working. If the pH is not optimal, you're not going to, the enzymes aren't going to be able to work properly. It's kind of like baking a cake. Like, you know, you can bake a cake at 200, but it's going to take forever to make, right? And you don't want to bake it too high, because obviously you'll burn it too. So you want that optimal level, if you will, of pH. So similar to temperature control, if you will. The pancreatic juices also contain water, and the one that we're going to concentrate on are enzymes. It also contains bicarbonate, which is what's going to help raise our pH, and this is why our pH of the pancreatic juice is about an 8. Remember, bicarbonate is a buffer. It's a alkaline chemical, and there's electrolytes in there as well. So the enzymes that we're going to concentrate on here are a few of them here are proteases, amylase, lipases, and nucleases. So proteases, these are just protein digestive enzymes, which is what we're going to look at in just a moment. Amylase is going to help produce or break down carbohydrates. Lipases help break down fats, and nucleases help break down nucleic acids. So you look at the pancreas, and this is an interesting organ because we can't live without the pancreas for this reason right here. These enzymes are able to break down all of our macronutrients, and we cannot say that about any other organ in our body. The pancreas produces all these enzymes that break down all of our proteins, fats, carbohydrates, and nucleic acids, which is quite unique in itself. So the one that we want to concentrate on, though, in just a moment are these proteases. Now, proteases are protein digesting enzymes. And I'm going to show you this picture right here. So here's your stomach, here's your duodenum, here's the pancreas. So these pancreatic enzymes or juices would be coming out this way. So imagine the pH of 8 coming out, trying to neutralize this area so that these enzymes can work properly here. Okay. Now, what happens is that when the enzymes here, these protease enzymes, come out, they're inactive, and they become active when they hit the duodenum. And so what happens here is this first one, trypsinogen here, is activated by an enzyme called enteropeptidase, which will then activate, and this trypsinogen becomes trypsin. This is an activated form of the enzyme that's ready to break down proteins. Trypsinogen is an inactive. Chymotrypsinogen is inactive. And so you can see that they're inactive, and they have to become active in order to work or break down proteins. So this first one, trypsinogen, being activated to trypsin because of this enzyme, enteropeptidase, is the enzyme that's activating this trypsin. Trypsin will then activate more trypsin. Trypsin will also activate chymotrypsin. Trypsin will also activate carboxypeptidase. And these three right here, as you can see right here, these are the proteases. These are the protein digesting enzymes, but they must become activated. And again, how does that happen? Trypsinogen becomes activated by enteropeptidase to become trypsin, and trypsin is the one responsible for activating the other two. Very important. If they are not activated, they can't break down proteins. Okay. Now, so the environment has to be right. You also have, because if let's say this pH isn't right, this enzyme right here can't activate these other enzymes at all, and so everything has to be optimal. It has to be within normal homeostatic condition, if you will. Okay, now we're just going to kind of summarize the macronutrient breakdown of our different types of macronutrients. Our first one is carbohydrates here. So you can see here it's telling you what's happening in each area of our body. So carbohydrates are polysaccharides. They're very large sugars and starches, and they have disaccharides here. That's fine too. Polysaccharides is our major one, which is starch. But amylase begins in the mouth, so we talked about that. Our pancreatic amylase as well from the pancreas that helps break down. And then once it gets down to the small intestinal tract, we have some other enzymes down here. Glucoamylase, lactase, maltase, sucrase. You can see they all end in ACE. These are all enzymes in the small intestinal tract which break it down to its simplest form, our micronutrients right here. So we want to take this macronutrient, we have to break it down to the micronutrient. And the reason for that is because we can't absorb it any other way, and our cells can't use it any other way. But our cells cannot use it as it is when we ingest it. It has to break it down to its simple form. It's like looking at a Lego house. You know, you want to build your own house, but you have to break down the current Lego house that you have in order to build it up. And so think about it that way. This is your big Lego house up here. We got to break it down to the individual Legos and use them. This is just showing you this breakdown. I'm not expecting you to know all of this stuff right here. Really just kind of the main points that you see right here. But you can just kind of see how the starch is up here and the pancreatic amylase is kind of working on it right here to break it down into more simple forms. The sugar then gets moved into the cells. Some of it's used in the cells, some of it's pumped out, and it's pumped out into the blood so it can circulate throughout your body to be used by other cells. And that's the the very simplest form I can tell you. Please don't read this and try to remember all those things. Not the point here. Now, what about proteins? So proteins are ingested pepsin is working on in the stomach there. We talked about pepsin. Remember, pepsinogen is converted to pepsin to help break down proteins, and then the protein gets broken down into large peptides, which the pancreatic enzymes that we were talking about that must be activated. The trypsin, chymotrypsin, and carboxypeptidase is going to activate, and then they start to work on even more of those proteins. And then the enzymes at the small intestinal tract that you see right here will break the proteins down into the very simplest form, which again is amino acids. Same thing here, very similar. Here's your pancreatic proteases, those three that we talked about. They're working on the proteins. These are all amino acids. The amino acids, or the protein, is broken down into these individual amino acids, which are pumped through. Some are used in the cell, and some are going into the circulatory system to go to other cells in the body. Fat digestion begins in your mouth. We have lipase, lingual lipase. The gastric phase has a little bit of lipase in there, very minor though. I didn't even mention that, I don't think, in my last, when we were over there in the stomach. But then emulsification, this is the big one right here, right? Bile is breaking down, emulsifying the fat. And then pancreatic lipase as well from the pancreas. And so our fat, which is essentially triglycerides, are broken down to monoglycerides and 3 fatty acids, since it's triglycerides. 1 monoglyceride and 3 fatty acids. That's what it would eventually break down to. That would be the micronutrients. So you better know, what does it start with, what does it end with, and what's getting us to that point, right? And again, just here's that picture that we covered earlier, and with the video, I'm not going to go through all that again. You can go back and look at that if you want. And the other thing that we look at is, with this, is lipoproteins, which is a lipid protein complex here. And so what this is a lipoprotein is a combination of triglycerides, cholesterol, phospholipids, and proteins. That's why I call it the lipoprotein. So a triglyceride, a cholesterol, and a phospholipid, these are the lipid part of it, or the fat part of it, and then you have your protein part. So a combination of these together creates what we call a lipoprotein. And so lipoproteins are needed for transportation of triglycerides, cholesterol, and phospholipids because they don't go well with water. We can't transport them in water. They have to somehow link up to something else in order for us to transport them in the bloodstream. So we call these lipoproteins. And it says increase in percentage of lipids and lipoproteins, the lower its density. And this is bad. Okay, so the increase in percentage of lipid means lower the density. If it has an increase in protein, that is a higher density. So we want more of the higher density versus the lower density. If we have more of the lower density, that means we have a higher percentage of fat. So a VLDL is very low density lipoprotein. So examples would be the chylomicrons. So if you go back to this, remember these are the chylomicrons right here. These are like the purest form of fat, right? That's a chylomicron, a triglyceride. These are bad for you. You don't want these lipoproteins, these VLDLs or LDL, low density lipoproteins. These are bad. This is your cholesterol panel. So you get a cholesterol panel back, you get these LDL versus HDL. HDL is rather good because it's got a lot of protein versus the fatty part of it. So you have more of that. That's a good sign to see. And the last one we really didn't talk about a whole lot here is nucleic acid, which is your DNA and RNA breakdown. So nucleic acids would be your DNA and RNA. The pancreatic ribonuclease and deoxyribonuclease are the enzymes that break down nucleic acids down to their simplest form, which we learned in a previous unit, which was the nucleotides, which consisted of a pentose sugar, a nitrogen base, and a phosphate. And again, we're not putting a big emphasis on that one in this particular unit, but it is part of our digestive process, if you will. And lastly is just quickly reviewing some blood flow in our digestive system. This is what we call the celiac trunk right here. We learned that back in general A&P, and it splits into 3 different blood vessels here. And we have one that's going to the stomach, which is your gastric blood vessel. We have one that's going to your liver, hepatic, and then one that's going over the spleen, which is your splenic. So the celiac trunk, it splits and the blood flow is going to the liver, the stomach, and the spleen. In this one, we're a little further down. In this one, we're looking at the superior mesenteric artery right here. We learned about that one in the digestive system in general A&P. And the superior mesenteric is going to provide blood supply to the pancreas, the whole entire small intestinal tract, and a good portion of the large intestinal tract as well. And further down, we have the inferior mesenteric artery. Let's see if I can find it here. Right here, inferior mesenteric. It's going further down here. And this is going to be supplying a good blood supply to the sigmoid colon right down here, as well as the superior rectum as well right here. So the inferior doesn't play as much of a role versus the superior, which plays a huge role in so much of that blood supply in the small and the large intestinal tract. And then our vein, our big one we have is a hepatic portal vein right here. It's a big thick one that's going into the liver right here. Why is this so important? Because we've got blood supply coming back from the stomach. We've got blood supply coming back from the pancreas right here in yellow. We have blood supply coming from the superior mesenteric artery. And we have, right here's the superior mesenteric, we have blood coming back from the inferior mesenteric artery as well. So all of this blood supply from all these organs is all coming back and running through the hepatic portal vein. And then it goes into the liver to be processed. So remember, the liver is processing our blood. It is filtering, detoxifying things, taking nutrients and waste products. And so this is kind of like border control right here, all right? So you got your hepatic portal vein, which is controlling what's getting into this liver here, and everything really runs right through this hepatic portal vein. So it's kind of the gateway into the liver, so to speak. So this concludes the digestive system. If you have any questions, let me know. Just please make sure you go back and review some of those finer details that